Good morning, students. I am Dr. Ruchi Jain, Professor in Sagar Institute of Research and Technology, Pharmacy, and today we will discuss the topic enzyme immobilization. As the name indicates, enzyme immobilization means immobilization of enzymes. Immobilization means we are not we are not allowing to move a enzyme. So here the enzyme immobilization, we are confining the enzyme molecules to a particular support over which a substrate is passed and converted to product. So enzyme immobilization may be defined as a process of confining the enzyme molecules. Confining means we are restricting the enzyme molecules to a solid support over which a substrate is passed and converted to a product. So what is an immobilized enzyme? An immobilized enzyme is one whose movement in the space has been restricted either completely or a small or to a small limited region. So enzyme immobilization technique in which we are restricting the enzyme molecule to a particular solid support. Support can be called as carrier and matrix also. So before moving further, we should know about the enzymes. What are enzymes? So enzymes are the molecules, they are proteinous in nature, they act as catalysts, they speed up the rate of reactions, they are found in every cell throughout our body, it lowers the activation energy, and generally enzymes are ending with the name ase means if they are acting on the lipid, then their name is lipase. So they are very specific in nature. Enzymes can lower the activation energy, means the energy which is required to convert substrate to the product. When we are using enzymes, the energy required is less uh, 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 in comparison to the reactions in which we are not using the enzymes. So enzyme can lower the activation energy. There are active sites in the enzymes which are binding to the substrates and we will get the products. The active sites generally occupy less than 5% of the total surface of the enzymes. They are very specific in shape and it depends upon the tertiary structure of the enzyme or protein. And whenever we change the shape of a protein, it will affect the active site and function of that enzyme. So active sites can be further divided into two types, binding site and a catalytic site. Binding site will help to choose the substrate and it will substrate will bind to that active sites. In the catalytic sites, the mainly the reactions will take place and the further substrate is converted into the product. So here is a diagram in which we are seeing that substrate is there, active sites in the enzymes are there. This is the active sites. Substrate can bind to the enzyme and it will form the enzyme substrate complex. And further, catalytic site will do its action and it will convert the substrate into the product. So, like that, end products will the form with the help of enzymes. So, we are seeing in these type of reactions that enzymes are not using in the reactions. So, why there is a need of enzyme immobilization? Because in the reactions enzymes are converting substrate into the product, but they are not using up in the reactions. So after the reactions, we can use that enzymes for further similar type of reactions. But when we are using enzymes, we this is we are not easily separate out these type of enzymes from the reactions. Hence, there is a need of enzyme immobilization so that we can easily separate out the enzymes after the reactions. And enzymes are very much costly. So after the reactions, if we can separate out the enzymes, then it will become a cost-effective uh, product or cost-effective reactions. So enzyme, by the help of enzyme immobilization, we have various types of advantages by which we are using enzyme to be immobilized. So once more, the enzyme immobilization may be defined as a process of confining the enzyme molecules to a solid support over which a substrate is passed and converted to a product. So why there is a need of enzyme immobilization? Because we can protect the enzyme from degradation and deactivation. We can reuse the enzymes for many reaction cycles and lowering the total production cost of the enzyme mediated reactions. We can easily stop the reaction by rapidly removing the enzyme from the reaction solution. We can enhance its stability. We can easily separate out the enzyme from the product, and further the product is not contaminated with the enzymes. There are some more advantages that we can increase the efficiency of enzymes, we can enhance the reproducibility of the process that are undertaking, we can use that enzyme continuously, less labor is required, we can save the capital cost and investment of the process, there is a minimum reaction time, less chance of contamination of the product, more stability of product will be formed, stable supply of product in the market we can maintain, and we can improve the process control. There is a high energy substrate ratio after the immobilized enzymes if we are using. So there are some disadvantages also. Advantages are more than disadvantages, but some disadvantages are that high cost of isolation, purification, recovery of active enzymes. There is industrial applications are limited in, and only a few industries are using immobilized enzymes. Sometimes the catalytic property of enzymes are reduced and completely lost after the immobilization process. Some enzymes become unstable after the immobilization, and if the heat is generated in the system, then it can it can inactivate the enzyme, and hence the enzymes are inactivated by heat generated in the system. So here the enzymes are confined to a solid support. So solid support here is a variety of solid supports are there. Solid supports are used to immobilize the enzyme by holding it permanently or temporarily for a short period of time, and it plays an important role in determining the performance of immobilized enzyme system. The matrix of support for immobilization of enzymes are grouped into the three major categories. They are natural polymers, synthetic polymer, or inorganic materials. They are further, and these carriers are inert, physically strong and stable. They should be physically strong and stable. They should be cost effective. They should be regenerable, and they should not inhibit the product formations. So ideal carrier matrix for the enzyme immobilization should be like this. There are further classification of carriers: inorganic carriers, organic natural carriers, and organic synthetic carriers. So inorganic carriers may be silicon dioxide, porous glass, silica, silicate, and xantonides. Organic natural carriers are cellulose derivative, DEA cellulose, CM cellulose, dextran, polysaccharide, agarose, starch, pectin, and chitosan. Organic synthetic carriers are polystyrene, polyvinyl acetate, acrylic polymers. Like that, many carriers are there for use of immobilization of enzymes. There are some natural polymers like alginate, chitosan, chitin, collagen, carrageenan, gelatin, cellulose, starch, pectins. In synthetic polymers, we have diethyl aminoethyl cellulose, polyvinyl chloride, UV activated polyethylene glycol, inorganic materials, zeolite, ceramics, diatomaceous earth, silica, glass, activated carbon, charcoal. All these are matrix which we are using in enzyme immobilization techniques. So before For the discussion of enzyme immobilization techniques, we should know about the carriers which are used to immobilize the enzymes. But what are the various techniques which we are using for the enzyme immobilizations are they are divided into two types, physically or chemically. In physical, we will see the adsorption, entrapment, and encapsulation type of techniques for immobilization of enzymes. In chemical, we will see the covalent bonding, or covalent bonding is further divided into support, covalent bonding, and cross-linking type of binding to immobilize the enzymes. It is a diagrammatically representation of various types of techniques for enzyme immobilization, like adsorption, in which enzymes are adsorbed to a particular support. In covalent bonding, there is a covalent bond formation between enzymes and support. In an encapsulation, enzymes are encapsulated into a particular capsules. In entrapment method, the enzymes are entrapped in a particular lattice formation of the gel or polymer. In cross-linking, there is a cross-linking formation between enzymes and support. So these techniques we will discuss one by one in the next class, and hope you liked the lecture and understood this lecture. Thank you.